Well, thank you very much for the gracious introduction. I'll tell you, it's lectures like these that I really enjoy. That is, giving lectures to students where you can actually talk about your own experiences and in a way, hopefully, that will be relevant to some of you. So what I'd like to do today, apart from tell you a little bit about my personal story, is make the argument that medicine and healthcare are at a big tipping point, and that scientific wellness is going to catalyze that tipping point. And by a tipping point, I mean medicine will really be reorganized in major ways. And in fact, to emphasize that point, in the US anyway, the first big tipping point in medicine came in 1910 when the Carnegie Institute mandated a report that, by Flexner that later became the Flexner Report. And that report looked at a chaotic scene of medical schools, none of which used science, and all of which practiced kind of trade mentality, both for dispensing healthcare and for teaching medical students. So his recommendations were really simple. One, bring science to healthcare, and two, bring science to the teaching of medical students. And at the time of the report, there were 165 medical schools in the US. 15 years after the report, there were 37. So that shows you the kind of reorganization that occurred. And what happened is, one, Johns Hopkins really took this report to heart, and it really pioneered this new modern medicine. And by embracing this tipping point, it placed itself in a leadership role. And I think the really interesting question to keep in mind is, who is going to embrace scientific wellness and place themselves in the leadership role for medicine of the 21st century? But let me explain all of that. So I grew up in a small town in Montana and went to a high school that had 136 students in it. But I had some exceptional experiences in Montana. One, my father was an electrical engineer who, in high school, had me take courses. He taught his men on systems engineering and circuit design. And so it profoundly changed how I thought about biology in a really interesting way. Number two, my granddad ran a summer geology camp for students and faculty from Yale, Princeton, Columbia, and Harvard. So I got to spend summers with those people. In fact, I almost became a geologist. But what I saw for the first time is real scientists and how they thought about things. And then the third experience, which was really transformational, as I was the first student ever from Montana to be one of the 40 Westinghouse Science Talent winners, and went by myself, first time I was out of the state of Montana, to Washington, D.C., and was utterly overwhelmed by how much smarter everyone was than I. And in fact, it almost made me rethink whether I should go into science or not. But fortunately, I then went to Caltech. I went to Hopkins, not to be a doctor, but to do medical research, to learn the fundamental information. And then I went back to Caltech and got a degree. I was at NIH at the wonderful Vietnam War time, because all the leaders in American medicine were at NIH. You got to meet the people who for the next 40 years were going to drive medicine in interesting ways, and then went back to Caltech as a professor, and that's where I'll take up the story. One of the things when I went back to Caltech I decided to do, and I told my chairman this, is I wanted my lab, on the one hand, to do technology development, and on the other hand, to do leading-edge biology, molecular immunology at that point in time. And I was enormously impressed with the complicated nature of both biology and the study of disease it was utterly clear we didn't have a way of talking about biological complexity, and later that came to be systems biology and systems thinking. But it was also clear that the tools at hand, the early emergence of molecular biology, biochemistry, cell biology, were utterly inadequate to this really complicated tool. And in fact, I remember reading in the spring of 1973 a book that really did impact me by Thomas Kuhn about paradigm changes in physics. So a paradigm change is an enormous reversal in how we think about something in science. And Kuhn pointed out two things. One, that paradigm changes are really hard to come up with because they require you to think outside the box. And we don't teach students how to think outside the box. And I would argue there are very powerful ways for doing that. But the second point he made that was really interesting is even if you show that you demonstrate the feasibility of a paradigm change, it can be very hard to get people to accept it because most scientists are conservative and they're really unwilling to give up what they learned early in their life. So those are all things to keep in mind, and we'll talk about them more. Anyway, I had the good fortune to be involved in a series of paradigm changes, and I tell you about these paradigm changes because, on the one hand, they were in part driven by dealing with biological complexity, but what they led to inexorably was this point of view I'm going to talk about for healthcare in the future, which is, as you heard, predictive, preventive, personalized, and participatory. So you heard about the first paradigm change. In a sense, I brought engineering to Caltech, and we started there developing the first 4 of the instruments that you heard about, and then later developed 2 additional ones at the University of Washington, and then finally in the Institute. But what I think was very interesting about this is I'd been at Caltech just 3 years when the chairman of biology came into my office and said, I urge you in the strongest possible term to give up all this technology development and just focus on your biology. And I said, nope, I wasn't going to do that. And then 25 years later, I asked him why he ever said that to me. And he said it was because the senior scientists in biology felt it was unseemly to have engineering in a department of biology. And he said, what they wanted me to do is move you to engineering. At least I didn't do that. So biology is really a good department. Good departments are often paralyzed by thinking very narrowly. So just keep that in mind. We'll talk about it again a little bit later. So what these instruments did, you heard already, actually. It led to high-throughput biology, making lots of measurements, and big data, and these were essential things for what I'm going to talk about later. But it led to one other really important conclusion, and that was the idea that scientists have an obligation to take their useful knowledge to society. So I remember in 1979 going into the president of Caltech and saying, look, I'm developing the first 4 of these instruments. A couple of them have really come quite far along. I want to commercialize these things. And this president of physicists said, the role of an academic institution is scholarship and education, not commercialization. I said, my view is it's transferring knowledge to society and we have an obligation to do it. So he said, okay, Caltech is not interested in that. You go do it on your own. I give you permission. So I went out in the next year to 19 instrument companies, and at the end of that, I was 0 for 19. And I'm not inarticulate about these things, but I was really depressed until a venture capitalist from San Francisco, Bill Bose, who became a very good friend, called me and said, look, I hear you've been unsuccessfully shopping this around. I'll give you $3 million and we'll start a company. How does that sound? Now, a venture capital company for Caltech at that time was reasonably undigestible. And it took a long time of finagling, but in the end, we did create Applied Biosystems, and that was necessary for the commercialization of the first 4 instruments. And let me say, commercialization for these kind of things is really critical, because what academics are really good at is conceptualization and proof of principle. What they don't have the resources to do is to make things robust and to make them high throughput. Those are all things where you need to borrow from society to create the instruments that can be what they want to do. And so we created one company for the first 4 instruments. For the inkjet DNA synthesizer, we created Rosetta, which developed it to a certain stage, and then sold it actually to Agilent, who uses that now for large-scale DNA synthesis, both of DNA and of arrays and things like that. And then the final instrument, we created the company NanoString for the an instrument that could detect single RNA molecules and has been very useful in a number of ways. So I think the idea that you owe it to society to transfer useful knowledge is really absolutely a critical kind of point. So one of the instruments, the automated DNA sequencer, got me invited to the first meeting ever held on the Human Genome Project. And I'll tell you, when I put my first 2 grants in to NIH on the automated DNA sequencer, I got some of the lowest priority scores I've ever gotten in my career. And I got comments like, graduate students can do this, we don't need instruments to do it. Or there was one comment that said, this is impossible. And you know, that's a tough comment to respond to. So anyway, so what I did was I got a foundation and a company, Monsanto, collectively to put in about $800,000, and that was what we needed to take the sequencer, the first 2 steps. And then after we'd done that, it was pretty easy to get money from NIH because it had already been proven. So the Human Genome Project was another interesting experience. 12 experts came to Santa Cruz in 1985 to talk about the merits of the Human Genome Project. And I remember 2 things really strikingly. One, we concluded that it was feasible, although at that time, very difficult. And number 2, we were split 6-6 on whether it was a good idea. And the people against it were fanatically against it. And a lot of the arguments centered around big and small science, and big science would destroy small science. They turn out to be incredibly complementary, obviously, in a lot of different ways. But the institution that was most opposed to the Human Genome Project is the National Institutes of Health. Absolutely incredible. And they turned around only when a National Academy committee unanimously endorsed the Genome Project in '89, and NIH saw it was going to happen and became an important part, obviously, of the Human Genome Project. So what did the Genome Project do for us? I think the major thing that it did, from my point of view, is it gave us all the genes, and by inference, most of the proteins. And for the first time, we could think about global holistic systems approaches to doing human biology. And we'll talk about some of those issues a little bit later on. The other thing the automated sequencer did for me was make me realize that to do it, we had to bring together engineering, chemistry, computer science, and molecular biology. And I realized that if you wanted to be a really powerful department of biology driving leading edge ideas, wouldn't it be nice to have that framed on a cross-disciplinary network with all different kinds of scientists who had learned to speak one another's languages and to work together effectively in teams? So in 89, I went to another president of Caltech and said, look, I have this great idea. I'd like to start a cross-disciplinary biology department at Caltech. And the president, who was an engineer, thought it was a great idea. The chemists loved it, the engineers loved it, the physicists were indifferent, and the biologists hated it. And I'll just leave it to you to, but that was something that I really wanted to do. So in 1992, Bill Gates made it possible for me to move to the University of Washington and set up the first such department. And what I'd planned to do was set up the cross-disciplinary framework and then build on top of it the systems biology. And the dean and I had really agreed on this, and that department was spectacularly successful. It developed the first two critical techniques in the then newly emerging field of proteomics, Rudy Abersol and John Yates. Phil Green developed the two key pieces of software for the Human Genome Project, assembly and quality control. Ger van den Ink developed an incredible multi-parameter high-speed cell sorter. We developed the inkjet synthesizer there, but serendipity is really a strange bedfellow. What happened at year 4 when the dean had promised to give me another floor to start building systems biology, he died in an avalanche in the Himalayas. And the next dean that came had a totally different view of science and medicine and where it should go. So I struggled with him for about 3 years, and then I decided nothing would ever happen with this guy. So I resigned and then went and set up what came to be called the Institute for Systems Biology. And there we really focused on systems approaches to both biology and disease. And I'll talk more about what kind of an institution this really was in just a second. But we also started to focus immediately on systems, that is global holistic approaches toward disease. And we were able to conceptualize two important ideas for healthcare in the future. So one was the systems application to disease, which we came to call systems medicine. And the second was this idea that healthcare should be predictive, preventive, personalized, and participatory. And we called that P4 healthcare. And I think what was really interesting then is at about this time, and I would say this is roughly 2012, something like that, I really had started thinking about how we could really bring P4 medicine to the healthcare system. And what we'll talk about later is really health is about two things. It's about wellness and it's about disease. Wellness is almost entirely ignored in almost all contemporary health systems. So we decided we would do an experiment on something that we called scientific wellness. And what that means is quantitative, high-throughput analysis wellness. And we'll talk about that in some kind of detail. And I'll just say, our year-long experiment in 2014 was so successful that led the next year to a company that commercialized with the intent of bringing scientific wellness to the consumer, and that's called Aerofail, and we'll talk about that very briefly. And this then positioned me in 2016 at where do I go next? And then what happened was really spectacular. The CEO of one of the largest nonprofit healthcare systems in the US, centered in Seattle, came to me and said, look, we would like the Institute to become our research arm and you are chief science officer. Are you interested? So we talked, and it turned out that this is an incredible opportunity, and I'll show you exactly what we're doing. But let me tell you that in the preceding 10 years, I had gone to 6 or 7 academic clinical medical centers making proposals that were a small fraction of what we'll talk about near the end of my lecture, and universally was unsuccessful. And that wasn't because a lot of people weren't interested in these systems medicine and P4 medicine ideas. It's because academic centers inevitably have silos with strong, powerful people who control their territory. And they control it both with regard to science and they control it with regard to resources. So they're very skeptical about outsiders come and starting new kinds of programs. And in every case, I went to my alma mater, and I was sure, since Hopkins had done the first tipping point back with the Flexner report, that they'd be willing to do the second. But Hopkins is just like all the other classic academic medical centers. So you have to ask yourself, in Sweden, what are your best medical centers like? What's the Karolinska like? And I have my own opinion, and maybe we can discuss that idea. Anyway, so this last paradigm change was the marriage of systems thinking with medicine. Okay, so let's talk then about the lessons that I learned from this. And I think there are three that I'll leave you with. One is this idea that every one of these paradigm changes met with enormous resistance. And some of them, like systems biology, there was really widespread resistance because people didn't believe it was real and so forth. Number two, which was really interesting, is I am totally convinced anytime you have a unique new idea, you have to create a new organizational structure. I don't think new ideas, big ideas, can flourish in old bureaucracies, almost never. And the final point I would say is, if you're going to do these kind of things, you need a lot of determined optimism, because you need to be able to look all these critics in the eye and say, nope, I'm going to do it anyway and try. And look, you ought to try. I mean, it's a good experiment, even if it fails, if you give it really a good push. So the Institute, I think, is a really unusual organization. Flat in administration, we can make decisions incredibly quick. We take these global holistic systems approaches to biology and disease. We've developed, as you'll see, a lot of technologies and a lot of analytic tools. We have a wonderful cross-disciplinary environment where a lot of us have learned the languages of the other disciplines and how to work effectively in teams. We really push big science ideas, that is, solving problems where you have to put together the talents of many different kinds of scientists so that you can attack them in a systems way, a global, holistic kind of way. That requires, obviously, critical strategic partnerships, and I'll talk about in a moment one of the most impressive strategic partnerships we ever had. And then finally, the Institute has been successful in transferring knowledge to society in a lot of ways, perhaps the most obvious of which is we've started 8 companies, and they collectively have brought back to the Institute tens of millions of dollars to do science. So it's been a really profitable, interactive kind of relationship. What did systems thinking contribute to systems medicine? I would say 4 things. One, the idea of dense dynamic data clouds, the idea humans are incredibly complicated, and to begin to understand that complexity, we need to make many different kinds of measurements. And of course, the idea is we can put all these measurements together and analyze it, and it will lead us to actionable possibilities for the individual to either improve wellness and or to avoid disease. And the dense dynamic data clouds work well in understanding wellness or in understanding disease, and we'll talk about both of those. And they're the essence of what a precision medicine should be, but in almost all countries, precision medicine is 90% genomics, and that is a serious mistake. The second thing we contributed is the idea of biological networks that are hierarchical in nature and operate at many, many different levels. And they're the information conduits that create human development, human physiology, human aging. And when they become disease-perturbed, they cause disease. And if you can distinguish key networks and their disease-perturbed nature from normal, you can begin to, 1, get fundamental insights into biological mechanisms. 2, you get terrific candidates for biomarker possibilities. And 3, you can begin to think about drug target candidates and so forth, and we'll show you some examples of that. We did do a lot of systems-driven, both technologies and strategies, and the technologies are straightforward kinds of things that we'll talk about. I mean, to give you an example, we've used now beautifully targeted proteomics in blood to get really effective biomarkers for lung cancer, for preterm birth, for PTSD, post-traumatic stress disorder, and for glioblastoma. And I think the techniques for doing biomarker discovery are utterly straightforward, but you have to use systems approaches. And I'd be glad to talk about what that means. And of course, the strategies are putting together one or more technologies with a platform that makes them really high throughput. Family genome sequencing is a great way in a single family to discover disease genes. And in fact, we used it in 42 affected families with bipolar to identify 125 genes that are involved in the bipolar process. But what I'm going to do in just a minute is talk about two of these technologies, third-generation sequencing and peptide protein capture agents. And one of the strategies, a new strategy for creating drugs, just to give you an idea of what some of these systems approaches are all about. With regard to data, we want it to be global, we want it to be dynamical, either temporal or spatial, we want it to be integrative, and we're really concerned about how you differentiate in big data signal from noise. And the noise comes from two places. One is the limitations of the technical measurements, and that's straightforward to deal with. But the noise that's really hard to deal with comes from the biologies that are irrelevant to what you're looking at. And there you need systems thinking to see how to actually subtract that out. So let me talk first about third-generation DNA sequencing. So it's single molecule sequencing. It uses nanopores. It uses electronic detection. And I'm now working with a company that has beautiful silicon chips that operate by virtue of conductance rather than induction. And what that simple fact does, it means, one, you've got greater resolution in what you can look at in that pore, and two, you can sequence 10,000 times faster than you can with ordinary protein nanopores and things like that. You can do very long DNA sequences. You can actually identify the epigenetic marks as they pass through the DNA that's being sequenced, the 16 or so chemical modifications of DNA. You can sequence, in the one instrument we're talking about, RNA every bit as easily as DNA, and of course you can sequence complete RNA molecules, since you can probably do hundreds of kb up to a megabase in terms of sequence, and if in fact you can prepare DNA fragments that are that large. But what is really exciting about this new device is we really begin to see how you can use it as a protein sequencing device. And proteins are really complicated because of editing, of alternative splicing, and of chemical modifications. And you'll be able to handle those at the single molecule level, large numbers, very rapidly sequenced. And of course, for silicon chips, what's great about them is you can parallelize them. So we really could conceive parallelizing it to the point you could do a human genome in 15 minutes. And I imagine in a 10-year period, the sequence cost will be under $100 or so. Second technology is peptide-based protein catalysis, pioneered by Jim Heath at Caltech, who actually is moving shortly to Caltech to join us. And the idea that Jim came up with here is you can synthesize a heterogeneous collection of 5-mer amino acids and circularize them and create a library of 10 to the 6th or 10 to the 7th. Then you can take a protein and you can use it to select low-binding monomers to that protein, and you can use a chemistry called click chemistry to join two peptides together to create in the proper three-dimensional orientation, a dimer or even a tetramer or a quatramer. Each mer you add, you get a half log to log increase in sensitivity. A dimer of the circular peptides is equivalent to the best affinities of really good monoclonal antibodies and so forth. And of course, what's really interesting about this, peptide protein-based chemistry is, one, these reagents are infinitely stable. So you could put them in envelopes, send them to Africa, the affinity would be unchanged. Number two, you can make the affinity as great as you want by adding additional kinds of mers, though I think for most cases dimers are going to be fine. And number three, they're digital. If you know what the two fibers are, plus the structure of the connecting chemistry, you can assemble everything in vitro with copper catalysis, so you can make large quantities. And it isn't great as a commercial thing because it'll be too easy to do, but you can, even more important, we can create, we can reduce enormously the cross-reactivity of these reagents by directing them against carefully selected peptide epitopes. And I don't know how many of you know, but monoclonal antibodies, when you look at complex mixtures, are plagued by cross-reactivities that you can't detect. And these, we can reduce those to a real minimum. And we see how we can scale these things up to do very large scale. And we've started a company called Indie Molecular that's beginning to do just exactly that now. We've demonstrated both in vitro diagnostics, in vivo diagnostics, and these are going to be incredible drugs, therapeutic reagents in the future. And we're actually working with Merck and immunotherapy to do some incredible proof of principle kinds of things there. So my prediction is in a 10 to 15 year period, these will replace monoclonal antibodies completely. See, what's really key about science is you always want to make bold predictions so people will remember who you are. And in some cases, they'll remember who you are because you failed, but that's okay, right? And the final thing I'm going to talk about is what Mike Tyers at University of Montreal. We're trying again to recruit him to ISB, and I think we're going to succeed. But in fact, he was all ready to come until we elected Trump. And he said, who would want to move to America with a president like that? And, you know, I couldn't disagree with him, but that's another story. So, so the idea that he came up with is he can synthesize 1 billion natural synthetic products extremely rapidly, and he can use yeast-type high-throughput screening to look for selected drug targets very rapidly. And the idea is really a clever one. You can take the metabolic enzymes from all the microbial genomes that have been done, and you can synthesize them and put them together in cassettes where you can put them in a YAC vector in cassettes of 4 or 5 or 6. And of course, the order of the enzymes that you place determines the scaffold and the structure of how the scaffold is decorated and so forth. And these, in theory, you could easily do 1 billion different drugs with this. He's synthesized 16,000 genes and done these kinds of playing. And what's interesting about these natural synthetic products is 60 to 70% of the drugs that exist on the market today are either natural synthetic products or derived from them. So this is the space you want to search for the new drug targets of the future. And what's interesting about this is you can do these things real time so that you could think about dealing with 2 infectious disease problems: emergent infections or drug-resistant microorganisms, TB and things like that, because you'll be able to identify drugs real time. And the cost of screening these things is somewhere between 1,000th and 10,000th of what it would cost ordinary pharma to do these. So all of these things started to converge, systems medicine and the digital revolution and big data, and an area I call connectivity, which is incredibly important area, but we're not going to have time to talk about it here. But what it let us do was define with greater clarity exactly what P4 medicine was about and how it differed from contemporary medicine. One, it's proactive, not reactive. Two, it's focused on an individual and not on a population. Three, it is about wellness and disease and not just disease. Four, it's about using these dense dynamic personal data clouds to be able to assess genetics and environment and how they collide within the individual. And fifth, and this is a really important point, I think the way pharma, for the most part, is doing clinical drug trials now is absolutely fatally flawed. So you take 20,000 patients and you give them cancer drug or placebo, you record the data, but you ask yourself, are each of these patients unique genetically? Are they unique environmentally? The answer is yes. And if you average disparate objects, what you always do is increase noise over signal. And pharma has really proven that in spades. So what would P4 medicine do? We take 20,000 individuals, each with their data cloud, and based on their individual data clouds, we'd stratify them according to properties we're interested in. For example, are you a responder or a non-responder to a given drug? And just to drive that home, here are the 10 most common drugs sold in the US, and the orange person is the reactor, and the blue people are the non-reactors. So in the best case, 1 in 4 works, and the worst case, 1 in 25 works. Look at the money we're wasting. Look at the morbidity we're causing. And when you do population-based drug screens, you have no idea how to separate the responders and the non-responders. P4 medicine gives that to you in a straightforward way right up front. So the two big themes, wellness and disease. And I'll argue that in US anyway, 96-7% of society's resources go to disease. Almost none goes to wellness. And I think what's going to happen with what I'm going to show you is there'll be a whole new industry in healthcare called scientific wellness. And there'll be the emergence of absolutely fascinating new kinds of companies, and we'll talk about some of those examples. So what about this whole idea of dense dynamic personal data clouds? They can do so many different things. I think this platform is really going to revolutionize the whole healthcare business. One, we can optimize wellness. Two, we can use it to follow disease and come to understand what's happening, its progression, and its nature. We can even use it, and as we'll show you in a moment, to see it at the very beginning. Three, we can use it to study clinical trials in really interesting ways and immediately stratify disease and individuals, immediately look at the cross-reactivities that might lead to inadvertent reactions of the drug. So you can do that very, very quickly. And of course, what's going to be interesting is we'll be able, with large populations, which we'll have soon, of individuals following them longitudinally with these dense dynamic data clouds, to see transitions from wellness to disease. We'll be able to get the markers that mark the initial transition point. And with network biology, we'll be able to get drugs that will reverse disease before they ever manifest themselves as phenotype, and that's the preventive medicine of the 21st century. And then I'll just say, without going into a lot of detail, N-of-1 experiments are absolutely going to revolutionize the drug industry in a lot of ways. And for example, metabolism, which I would argue is really in the dark ages, the only way to understand metabolism is with large numbers of assembled N-of-1 experiments. And again, we can talk about that later on. So when, as I said earlier, I decided I would like to attack scientific wellness, again, a great story. I went to Francis Collins at NIH and said, look, we're going to do this great project on scientific wellness, and I'd like the National Institutes of Health to help on this endeavor. And he said, nope, we're not interested. This doesn't have anything to do with our mission, which is related to disease. So I said, okay. And then I went home and raised $2.5 million from philanthropy to be able to do the project I'm going to talk about with you right now. So before I do that, let me say there are a lot of studies that have looked at the determinants of health, shown that genetics are about 30%, environment's about 60%, and healthcare is about 10%. And of course, the really critical thing is it's these dense dynamic data clouds that let you assess the two major components, that is genetics and environment in this endeavor. So what we did was complete genome sequence of each of 108 individuals. So I got 108 of my friends to agree to subject themselves to dense dynamic data clouds for a year. And about 50% went in incredibly skeptical about whether this was going to be a worthwhile, but it was free for most of them anyway. So they agreed to do it. Complete genome sequence, we did blood, urine, saliva draws 3 times a month. That gave us about 200 clinical chemistry components and 1,000 metabolites, and we looked at 400 proteins initially, roughly. We did the microbiome every 3 months as well. We did digital measurements using a Fitbit to assess activity and quality of sleep, and did blood pressure and pulse and all those other things. And of course, for each individual, you got this data cloud of billions of data points, and we showed that you could indeed analyze these and lead to actionable possibilities that could let you either improve health and or avoid disease. And let me say, we got the actionable possibilities initially from the data, and we got close to 1,000 of those, including all the different data types we had. Since we've started gathering the data, these actionable possibilities have increased exponentially, and they've increased in sophistication as you integrate together different kinds of data types. And we can talk about that. So what we did then is every month we had coaches call up the pioneers and give them their 2 or 3 top actionable possibilities that they could use to change their life. And I just want to emphasize that these scientific wellness coaches are absolutely critical. They both can explain the actionable possibilities, and they're good psychologists that can talk to the pioneers and come to understand what their healthcare objectives are, placing the actionable possibilities in that context. And they are what is absolutely critical for long-term recruitment of the pioneers. We have people that have been doing this now for almost 4 years. So it's been a really exciting story. The first blood draw we ever took, 96% nutritional abnormalities, 68% inflammation, and so forth. And we were able to correct these in incredible kinds of ways. One of the conclusions that they came to is almost all of the 100 weren't nearly as well as they thought. In fact, the average person was probably at the bottom of a wellness staircase, and it was the actionable possibilities that elevated you up. And what we wanted to do is get people to Eric Topol's Welterly, 80 or 90-year-olds that have never been sick a day in their life, never taken a drug, and they go into their 90s mentally and physically alert. So we think with these actionable possibilities, ordinary people can be elevated to that, but it means this is, in fact, a journey of a lifetime. And my vision is we'll follow people from conception to the end, letting them optimize continually their scientific wellness. Aerovale was the company we set up to do this, and we've a business plan now that will, by 2020, have more than 800,000 individuals that will let us look at these incredible kinds of transitions. And ISB and Aerovale are working together on the analysis of all of these different kinds of data. So I'm going to talk just very briefly about the data kinds of types that we got. Three major observations, roughly 6,000 statistical correlations, 4,000 from static data, 2,000 from dynamical data. They're almost completely different kinds of correlations. And they lead to really interesting things with regard to drug companies. Number two, we showed that we can estimate genetic risk for almost 100 different diseases now. And we show that in 21 of those cases, the genetic risk correlates beautifully with the disease phenotype and so forth. And we've got terrific state transitions for wellness and disease that allow us to follow the kinds of transitions. Now, because we're getting late, I'm not going to talk about any of the data, but if anybody's interested, I'd be glad to talk about it later. But it is some of the most spectacular data that I've ever seen. And we can do all sorts of new things with it, but we'll talk about that later on, because I want to get to the very last part, which shows you that this is an example of a diagnosed pancreatic cancer. We have all sorts of blood draws beforehand, and what we've been able to demonstrate is a single protein that is an incredible outlier that increases with all four of those blood draws right up to the diagnosis. So we have the way of getting powerful diagnostics, and this is not proof. It's just gives you a really good candidate. But if we can detect cancer really early, we want to use immunotherapy to eliminate the small nests of cancer cells. So that's really an exciting idea. So let's talk then about the affiliation that I talked about before. So it lets me do one, translational pillars. We'll talk about those. Two, we're going to do education of physicians and of patients. Three, we're going to create a big technology platform. And four, we're going out to other large healthcare systems, trying to recruit them into scientific wellness. And I won't have time to talk about any of those. Providence is a $20 billion operation a year. 7 states serve 50 hospitals, 5 million patients a year, 30 million electronic medical records, so we can do incredible things. So on the clinical trials, the translational pillars, ISB, Providence, Aerovale, and a company we set up to extract from Epic and these intractable clinical databases, anonymize patient records and put them in the cloud so they can be queried appropriately, opens up the possibility of a whole series of translational pillars. For all, we're going to use these systems-driven technologies and strategies as relevant, and of course, the dense dynamic personal data clouds. We actually have funding for the first 2 of these. Providence is doing 1,000 of its employees with Aerovale over a 3-year period to compare against 1,000 controls. 1, to show how much improvement in health occurs. 2, to show the enormous economic savings that occur, and that's critical, obviously. Alzheimer's, we're taking a systems approach. We've got a 36-point regimen that in preliminary experiments beautifully reverses the earliest transitions of Alzheimer's back to completely normal phenotypes. So we're going to do a big clinical trial, and I think within 3 to 5 years, we'll have the diagnostic markers from the blood to pick out the earliest stage of Alzheimer's and the ability to reverse it at that time. And a final point is the cost of these things is really enormous. It's about 4,000 or 5,000 at this point in time. What is happening is that there's an exponential Moore's law decline in the cost of these things. And I think really within a period of 10 years, the cost will be $100 or so. And we'll probably have a tricorder. You'll keep at home, and it'll take a drop of blood and do 6,000 measurements, and that'll tell us everything that we need to know. And we know how to do this. We just haven't done it yet. So anyway, that'll get in the insurance companies, and that will let us bring it to the poor as well as to the rich. So where are we? I think you can see we're at a healthcare that's predictive, preventive, personalized, and participatory. These dynamic data clouds are transformational in assessing genetic and environmental information for both wellness and disease. It's absolutely clear that we'll be able to optimize wellness. We'll be able to get these transitions and reverse them early. We're in a good position to start arguing scientific wellness ought to be a lifetime journey. The costs are going to come down. We will start a whole new sector of healthcare on scientific wellness. I think the data will transform how the healthcare industry operates. And of course, if we can bring the price down, we can bring scientific wellness to the poor countries as well as the rich countries of the world, and that'll lead to a democratization of wellness that is inconceivable even before. So the final point that I'd like to make is mostly for the students, although I think some of the faculty might well adhere to this too. So in my experience, if you plot success in the y-axis against time in the x-axis, for most people, you kind of get a bell-shaped curve. You go up to a maximum, and then you kind of fade away. But the way you can avoid the down phase is if somewhere near the top, you change what you're doing, you become insecure, you learn again, you get excited again, then you're going to have another ascending curve. And I think there's no limit to how many times you can do that. So you could even think about being immortal scientifically. But anyway, I strongly urge that you think about the idea that it is really healthy to change your field every 10 to 15 kind of years. I've done that, and it's been marvelous for me. So 1, science should be fun. 2, you ought to attack really big problems in science. 3, it's critical to work with the very best people you can. And 4, always remember to be determinedly optimistic. Thank you.